We're Pictura Bio, like I said. My name is Chris Stanfill. I'm the Chief Business Officer, and we actually use machine learning to do pathogen detection in vitro diagnostics. We were spun out of University of Oxford back in 2021, and we like to describe our technology as facial recognition for bugs. We do pathogen detection in 1 minute by reducing the complexity using software. And so we go from the traditional method of asking, do you have X or do you have Y, to basically going, what do you have when we do our tests? And so we feel the world needs instant point of care testing right now that can find anything. So the traditional way that we've all gone through having lab tests done is centralized lab testing. And it really can't provide actionable results when the doctor is with the patient, so someone can be tested and treated and then sent on their way. It doesn't have the speed to provide that, and that lack of speed results in patients queuing up, like we all experienced with COVID, or waiting hours or days to get your lab results, which they usually say, we'll call you if it's abnormal, and then in parentheses, it's actually, or if we forget. So there are POC tests on the market right now that try to address this, but the matter is they're just not fast enough yet. A 10 to 15 minute test just isn't fast enough because most appointments don't last that long. And so at Pictura, we feel we have the solution because we provide lab quality tests in 1 minute at point of care when the patient is still with the physician. And so our technology is a connected ecosystem with 3 different pieces of tech. Our first, we call it PIC-ID. And what this is a universal labeling reagent that you add to the sample, and it's what makes the pathogens visible to a microscope. And then the second component is our instrument, which we call the Vista Reader. This is the instrument that actually runs the test and then captures the images of the sample via an onboard microscope. And then lastly, the machine learning part is we have a neural network that actually analyzes the images and then identifies all the pathogens that are present and differentiates in between them. So how it works is basically you swab a patient, just like for respiratory infection, you swab a patient like we've all had done many times now, and you put your sample in with our PIC-ID reagent. And what happens is instantaneously everything in that sample is labeled with the fluorescent pattern, completely non-specifically. Then when you put your cartridge into the instrument, it takes a series of digital images automatically and then shows that to the neural networks that are on board the PC on the computer. And what the AI machine learning does after the image processing, it clips out the images that it thinks are viruses and then presents it to a neural network to do the identification. I'm going to explain more about that on the next slide. So that process we call is classification, and how it works is we have a convolutional neural network and we train network models to recognize different pathogens we bring to it and show it known positive samples. So it learns what a positive sample for one of these pathogens looks like. And then, like I mentioned before, when the AI is looking at the image, what it does is it identifies these like blobs and it clips them out if it thinks it might be a pathogen. And then all of these little clipped out images are shown to these neural networks that have been trained to identify a pathogen. And they each kind of look at it and flag anything they see as being that pathogen they were trained on. Then we convert this into a positive or negative result for each pathogen on our panel. And so this kind of was all developed during COVID. So our preliminary data is on COVID, and we're got about 95% specificity at the species level, so positive or negative. And then we can also discern different variants of COVID in this case, but it's much more difficult to do that. So you lose a little bit of the fidelity and the sensitivity. And so we're working on bringing that up, but the early data we've seen on it is very promising. And so we have 4 key differentiators of our technology versus the other POC point of care technologies that are already on the market. Number one is our speed, is we provide a multiplex result in 1 minute. So 5 different pathogens still only take 1 minute. One pathogen would take 1 minute. So highly multiplexed, very fast. We use a universal consumable or kit. Remember when I explained that the fluorescent labeling is completely non-specific, it labels everything in the sample. That allows us to have just one kit. And then when we couple that with the fact that we use the machine learning to add specificity to our tests, well, you put those together, and when we launch a new test, we actually push it out via software update. Then you combine that with your kit that's already in your lab, and you can just run the new test. You don't have to order a new kit. And then lastly, we have a public health application where, since the sample we analyze is really an image of the sample, you can look back at any test that's ever been done on our platform and retest that old sample with a new test just by reanalyzing the images. So this could give public health officials a window into when a new pathogen first showed up in the population. And many common infections have diagnostic tests that go with them. And the incidence of things like respiratory infections, which every person on earth gets 2 to 4 of these a year, urinary tract infections, or new STI cases, they'll support a very broad base, installed base at places like retail healthcare, like Minute Clinics or emergency departments, long-term care homes, which was a problem during COVID, being able to do testing there. And so the first market we're addressing, as I've kind of alluded to, is the respiratory infection market. And we're using a multiplex respiratory panel for that. And we broke it into 3 phases, and we're just finishing phase 1 now, where we've built our demo system and we've collected the requirements to build our alpha system. So by the middle of 2025, we plan to have built and field tested our alpha system, and we'll do our respiratory panel internal validation and then be collecting feedback from the market so we can set the requirements for our beta system. And then the next year, we'll build and field test our beta system and begin designing our clinical trials in anticipation of kicking those off shortly thereafter. And so to date, so far, we have several partnerships with both primary and secondary care providers in the UK. We actually have a clinical trial ongoing right now at Portsmouth University Hospital, where we're actually swabbing patients when they come to the emergency room and collecting an extra sample so we can compare against the sample that they're going to then have PCR'd. We also have several sources of non-dilutive funding. We just finished up a UK SMART grant to do proof of concept. And then in the next 2 months, we're going to deliver on a Ministry of Defense contract where we provide an instrument and assay for point of care testing on the front line. And so where we're going is have almost finished setting up our US subsidiary with the goal of having it set up. Most likely it's going to be at Aquilius to expand that clinical trial that's at the University of Portsmouth Hospital that I mentioned, expand that to the US. And we've actually already spoken to several researchers at UCSD. And so far, there's been some interest in further conversations on that. Then mid-2025, we'll have our alpha system followed by the beta system a year later, and then heading into clinical validation in anticipation of our US regulatory filing in 2029. And so in this round, we're looking to raise a total investment of $6 million. We feel we have a track record of proving that we meet our funding milestones. We have the right team to do it. And we have a flexible kind of business model where we can respond as new potential applications arise. But right now, we're very confident that respiratory is the place to start. And with that, I'd be happy to answer any questions you guys have. Thank you so much for your time.
Hi, this is Pilgrim from Yatiri Bio. Thanks for the talk. That was really good. My question is, if a new pathogen comes up, how quick is it to train the model and have something ready that's added to your kit?
So in order to train a model, we need positive samples. So all we would need is some positive samples from a public health lab, and it would take us about a week to have a new test that we could then push to a deployed instrument via software update. Now that would be for emergency use. It's not going to be validated through the FDA, obviously.
Hello, this is Sean. I have a question. So then you have to adopt your test, right? Because your model is trained based on your test. Different test, although the passengers may be the same, going to have different results. We're going to adopt the whole thing, the whole package, not just your picture bio, but also the whole test. Is that right?
I'm sorry, the mic was fading in and out. I didn't catch your whole question. Could you repeat it?
Can you hear me now?
Yes.
I'm just wondering, so since you build your model based on the test you are using, we have to adopt the whole package from the test to all the way to the readout. Is that right?
Right. We train with samples that are tested via another method, probably PCR. And then you do need our entire instrument and system in order to get a test result. It's not like an at-home or usage case or anything like that. I hope I answered your question.
Okay. Thank you, Chris. We really appreciate you presenting today.